Another very important experiment is Hershey and Chase experiment, also called as the Blender's experiment. This experiment proved that undoubtedly DNA is the genetic material, and it is an important experiment which was performed by Hershey and Chase in 1952. Now, for their experiment, they took a bacteriophage. Bacteriophage is a virus that infects the bacteria. Now, if we study the structure of a bacteriophage, the bacteriophage consists of a capsule that is known as the capsid, and this capsid is made up of protein. And the protein, that capsid, actually protects the genetic material which is present inside. So the nucleic acid, that is DNA, is the genetic material of a bacteriophage, which is very well protected with the help of a proteinaceous capsule known as the capsid. Then it has a collar and the tail fibers which help in the attachment of this virus onto the bacteria. Now, when this bacteriophage attaches itself to the bacteria, it will transfer its genetic material inside the bacterial cell, and the bacterial cell DNA will be modified, and as a result, the virus takes over the bacteria. Now, using this principle, Hershey and Chase used a bacteria that was T2 bacteria, and it was used to infect E. coli bacteria. So the T2 bacteriophage was utilized to infect E. coli bacteria. Now, for this purpose, this bacteriophage T2 was grown in a radioactive medium consisting of an isotope of phosphorus, that is P32, and an isotope of sulfur, that is S35. Now, this phosphorus was used because phosphorus is found in DNA, so the DNA was labeled with radioactive phosphorus, and the protein code was labeled with sulfur because the protein has sulfur content in it. So therefore, now we had two strains of bacteriophage, that is one strain that was having a labeled DNA, that is radioactive labeled DNA, and the other one had radioactive labeled protein code. So the protein code, which was the capsid, had radioactive S35 labeling, whereas the protein code, whereas the DNA, that is the nuclear material, was labeled as phosphorus P32. These two were grown, and the two were allowed to infect the bacterial cell. Now, this is the bacterial cell, and it was allowed. And also remember, when the bacteriophage attaches itself to the bacterial cell, it will transfer only the genetic material inside. So when these two were made to attach itself to the bacterial cell, one had the protein code which was labeled, I'll make it with blue, and the other had the genetic material. Now, the protein code will remain at the top and it will not enter whereas the DNA will enter into the bacterial cell. This is the DNA which is not affected, and here that is not radioactive over there, and here it is. This is radioactive labeled P32 DNA, and here it is the protein coat which is remaining outside. Now when this culture was grown and it was before the lysis or the breaking of the bacterial cell, the two were blended, put into a centrifugation, and two different regions were obtained. That is, the denser ones settled down, that is the particles or the bacterial cell which were heavy, settled at the bottom, whereas the lighter cell form the supernatant. Further analysis showed that the heavy bacterial cell had radioactive DNA, whereas the lighter bacterial cell region had the protein coat, which did not enter into the bacterial cell. Now, this was further used to infect the bacteria, and it was found that the DNA caused the change in the genome of the E. coli, whereas the protein coat did not cause any change in the bacterial genome because it did not enter inside the bacterial cell. So this experiment proved that it is the DNA that is the genetic material and is responsible for the transmission of the traits and is the hereditary substance.